Hello, everyone. In this lecture, I will be talking about at the epigenome from the transcription factors and histone modifications point of view. So in the previous few lectures, we've already learned a lot about the epigenome from Dr. David Albo's lecture and Nathan Sheffield's lecture for the past two lectures. And also, Dr. Gerten and Dr. Bekianov talked about transcriptome and the determinants of different transcription programs, as well as some analytical tools for analyze transcriptome data. So they are all related. In this lecture, we will continue to be talking about the epigenome. So first, I will give you an overview, although you've already heard about epigenome for the past two lectures. And in this lecture, I'll be focusing on one technique for measuring the chromatin epigenome, the so-called ChIP-seq. I will briefly introduce the technique about ChIP-seq and briefly go over the data analysis for ChIP-seq and a little bit about the future perspectives. So first, let's have another overview of the epigenome. So according to the definition from the National Human Genome Research Institute, the epigenome is a multitude of chemical compounds that can tell the genome what to do. The epigenome is made up of chemical compounds and proteins that can attach to DNA and direct such actions as turning genes on or off, controlling the production of proteins in particular cells. As we all know that the human and the mammalian eukaryotic cells, the double-strand DNA is wrapped around a core of 8 histone molecules to form the structure called the nucleosome. And the nucleosomes and the DNA form a structure like a bead on the string. And after a few rounds of additional packaging, they form a very condensed structure of the chromatin. So in one cell nucleus, say a human cell, the entire DNA, if we extend it to a linear molecule, the size is about 2 meters long. But then in the cell nucleus, it can really condense into a very small size of a cell nucleus. So this packaging process is done by an interaction between the DNA molecule and the various type of proteins. And these proteins, as I mentioned, includes the chromatin proteins such as histones, as well as other proteins that are directly or indirectly associated with the DNA, including transcription factors. And all of these protein factors can be subject to some chemical modifications. And some chemical modification like methylation can directly modify on DNA, such as DNA methylation. So all of these factors on top of DNA sequence itself are epigenome factors, and all of the factors adding up together is made up the epigenome. So the factors or marks of the epigenome, like I just mentioned from that last slide, that includes DNA methylation and histone marks. Histone marks includes histone covalent modification that I will go through in the next slides, as well as histone variants. It's a variance type of histone with a certain amino acids difference from the original histone molecule. Also, transcription factors is a group of very important proteins that directly interact with DNA that with the main function of regulating the gene expression program or transcription. And chromatin regulators is another group of important marks in the epigenome that includes, but not limited to histone modifying enzymes, including chromatin writers, histone mark writers, readers, and erasers, and also chromatin remodeling complexes like SWI/SNF complexes. So let's talk a little more about histone marks. So in each nucleosome, there is a core particle that consists of 2 copies of each of the 4 core histones. They are H2A, H2B, H3, and H4. So each histone will have 2 copies. So they form a octamer to as the nucleosome core particle. And then the DNA sequence is wrapped around this core particle for 1.5 rounds, or about 150, or precisely 146 base pairs. And then on the tail of the histone molecules, these protein molecules, some of the amino acids on the tails can be subject to different types of covalent chemical modifications. And such modifications can include methylations, acetylations, phosphorylations, ubiquitinations, and so on and so forth, and many other type of, I think there are nearly dozens or nearly 100 different type of histone modification that have been identified. I think Dr. Obo have given a more detailed overview of all of these modifications. So among those histone modifications, there are 6 of them that are most commonly used or widely studied. So I'll briefly mention the notation about the histone modification. So histone modification is notated as H3K27AC, which means that this is a histone modification at histone H3 molecule on the tail and the 27th amino acid, which is a lysine, and this modification is an acetylation. So H3K27AC means histone H3 at lysine 27 acetylation. And methylation, they can have monomethylation, dimethylation, and trimethylation. So noted as ME1, ME2, and ME3. So these 6 modifications, including 1 acetylation, 1 monomethylation, and 4 trimethylations, are the we most commonly see in modifications. As we see, they occurred, all of them occurred on lysine amino acids. So I'll mention that in more details in the next few slides. So in addition to histone modifications, there are histone variants. Some commonly seen one include H2AZ, H3.3. All of them have different associations or functions in the genome associated with gene expression. So in general, histone modifications are implicated in influencing gene expression. So generally, to study the association between these histone modification marks and their associations with gene expression, so this is a work published in many years ago that I did, in which I did the analysis, in which we analyzed the ChIP-seq profiles of nearly 40 different types of modifications, histone modifications in human T cells. And we try to associate these modification signals with the gene expression. So as we can see on the gene promoter regions, we on the left plot, we separate all the gene promoters into the group that have each modification and the group that doesn't have this modification. And then we compare the expression level of the genes between these 2 groups. As we can see in this plot, most of the histone modifications will have a elevated role or association with gene expression, whereas some other modifications will have a reverse correlation, meaning that having this modification will have a lower expression than the genes that do not have this histone modification. That's directly affecting the gene expression if they're modification at promoter regions. Whereas another important group of DNA regulatory elements are enhancers. And in this, we also look at the DNA's hypersensitive sites, the open chromatin regions. And we want to see how many of the enhancers regions are associated with each type of histone modification. As we can see, they have a various level of association at that in which indeed we'll talk about mentioned later, including H3K4 monomethylation and H3K27 acetylation. They are mentioned to be important marks with histone with enhancers. So histone modifications are associated with promoters and enhancers. So for these 6 common histone marks, I think this is what we will see in again and again. So we want to summarize the information or the functional annotation of these 6 common histone marks. So H3K4 trimethylation is usually marked at the promoter regions, not necessarily meaning that the gene will be active. In fact, H3K4 trimethylation can mark both poised and active promoters. And there is an interesting type of histone modification combination, or a bivalent, so-called bivalent domain, meaning that some histones can be marked by both H3K4 trimethylation and H3K27 trimethylation. They can mark the so-called bivalent or poised promoters. So because H3K4 trimethylation have a potential of activating gene expression, whereas H3K27 trimethylation will have a role of repressing gene expression. So putting together, these will form a bivalence domain. H3K36 trimethylation marks the transcribed gene body, meaning that this gene is usually actively transcribed or actively expressed. For H3K4 monomethylation and H3K27 acetylation, they are both marked at enhancer regions. As you can see here, they have a difference that K4 monomethylation can mark both active and poised enhancers. And active enhancer, H3K27 acetylation is the single mark that clearly mark active enhancers. So I just mentioned that H3K27 trimethylation is associated with repressive chromatin. So they are associated with silence or repressed genes. So they are polycomb repressive regions across the genome. They are associated also with polycomb. And the last from the 6 modifications are H3K9 trimethylation. It's associated with heterochromatin that's really closed and silenced the chromatin region. So let me talk a little more about the H3K4 trimethylation and the K27 trimethylation bivalent domain. So this type of bivalent domain are most commonly seen in stem cells, embryonic stem cells or hematopoietic stem cell, or any type of stem cell that can have the potential of differentiating into different other cell types. So in the poised states, the domains can have both K4 trimethylation and K27 trimethylation marks, which means that they are poised and have the potential to be either activated or further repressed. So after cell differentiation, these bivalent domain can have different fates. If after the differentiation, these particular regions the genes are no longer needed to be activated, or even the potential don't need to keep, don't have to be kept, so they can add more, have a higher level of K27 trimethylation, and the K4 trimethylation can be removed. So it's a further meaning that these regions can be further repressed. And of course, they can still remain as bivalent domain by keeping both K4 and K27 trimethylations. And the most interesting group is that they can be induced with the H3K27 trimethylation gone, by keeping K4 trimethylation and adding other activated marks, including acetylation, so that these regions of chromatin or the genes associated with these regions of chromatin can be induced and activated. So by having this bivalent domain, bivalent mark of histone modifications, they can give the certain regions in the genome or certain genes different fate during cell differentiation. So this is a very interesting type of epigenetic regulation for gene expression. So, of course, there are a lot of studies and a lot of debates about the actual functional role of these histone marks or histone modifications. I would say, although there are various evidence that could suggest the inducing or the histone marks can be the cause of gene activation, but many of the evidence is not that strong. So that in most cases we are just observing the association or correlation between these epigenetic marks or histone modification marks with gene expression. But we should just keep in mind that they are not necessarily means causation. So now, next, let's talk about another group of important epigenome marks, transcription factors. So transcription factors is a type of protein that can usually directly interact with certain regions in the DNA or directly bind at certain regions of the DNA that have the direct role of activating transcription or of the genes in nearby genes. So a transcription factor, in order to have this function, they usually have two different domains in their protein structure. So one is the effector domains. So the effector domains is those that have this to play the role in the transcriptional activation, in which they can regulate this transcription factor activity by binding, receiving signal from the ligand. So these domains can be a ligand binding domain, or they can mediate protein-protein interactions by interacting with other protein molecules, or they can have enzymatic activities to add or remove histone modifications or in general chromatin post-translational modifications on the other type of proteins. And the other important functional domain on transcription factors is the DNA binding domain. Though these domains can recognize specific DNA sequences along the DNA sequence. And as this diagram shows an example, these transcription factor has 5 zinc fingers. So each zinc finger domain can recognize certain DNA sequence, and they form the very specific DNA binding sites. So for each transcription factor, they usually can recognize a specific type of DNA sequence. And these DNA sequence that a transcription factor recognize can usually be represented by such a sequence logo that I will mention a little bit more later, which means the frequency or likely the possibility of having certain bases at certain position across the DNA molecule that represents certain sequence, their specificity and their ability to be interacting with the DNA binding domains. So just a brief overview of a characterization of transcription factors. In terms of its structure, they need to have effector domain and DNA binding domains. And the function of the transcription factor includes, of course, activating cell type specific expression, activating or repressing, or in general, regulating cell type specific gene expression. And they do that by directly binding to the DNA sequence or what we call DNA sequence motifs, reoccurring sequences that a certain transcription factor can bind. Then across the entire genome, each type of transcription factor can have many different binding sites across the whole genome. And they, by binding at different regions across the genome, they can regulate many different target genes. And they can also interact with other cofactors and other protein molecules to play a role in the regulating the whole chromatin structure and the gene expression program. So many transcription factors exhibit tissue-specific or cell type-specific expression patterns. And this plot is from a summary, it's from a review paper recently about transcription factors. They plot the gene expression level of 1,500 human transcription factors across 37 different human tissues. As we can see, for any given tissue type, there are only a small fraction of all the transcription factors that are highly expressed. And if we look at each column, we can see each transcription factor, they usually are only highly expressed and functional in a subsets of cell types. So that means the cell type specificity of the transcription factors. And by this cell type specificity of transcription factors is one of the very important determinants of the cell type specific gene expression profile that defines the cell identity. So I also wanted to mention that a transcription factor doesn't have to have a very high expression level to be functional. Because if you can imagine, the size of the cell nucleus compared to the entire cell is relatively small, and compared to some other proteins that need to form the structure of important components in the cell, the transcription factor only needs to have certain amount of protein copies inside the cell nucleus. As long as they can bind to the certain regions in the DNA, they can be functional. They can activate their target gene. So they don't have to have a very high expression level. So that's another reason that the transcription factor is very I would say that another reason that transcription factor is interesting to it's very interesting to study, and it's not easy to be completely knocked down because they only needs a very relatively low expression level to have the full function. Then I want to talk a little more about the sequence logo that we always see to represent a transcription factor binding motif. So a sequence logo of such shape of in each position, they have the 4 bases ATGC of different heights represents the whole DNA motif. Such plot, so-called sequence logo, is derived from position weight matrix. So the position weight matrix is one type of representation of recurrent DNA sequence binding transcription factor binding DNA sequence or DNA sequence motifs. So for example, if we collected the transcription factor binding sites and found that this transcription factor can bind to a different type of DNA sequence across the genome. And this is an example of 10 DNA sequence fragments that some transcription factor can bind. So how to derive a position weight matrix to represent the DNA motif. So first we align these sequence and we generate this matrix simply by counting the occurrence or the frequency of each base pair, each basis in each position. So at the first position, there are 3A, 2C, 1G, and 4T, and so on and so forth. And as we can see in the fourth position, all of them are consistently to be G, and the fifth position, all of them are T. So this we simply do a count of all the bases in each to form this matrix. And then we convert it to the frequency. So on each position, the frequency add up to 1, right? So the 100%, all of them are G, and 100% of them are T. And that's such represented at each of these 9 positions. So, and this matrix is the so-called position weight matrix. At each position, each base will have a certain weight of representing the frequency that this base occurring at this position. Is very intuitive. And then we just plot each base in such a sequence logo plot. At each base, the height of each base at each position, the height of each base from A, C, G, and T will be proportional to their relative, to their corresponding frequencies that this base represents. And after that, the entire height of this base it will be represented as the information content at this base. As we can see, the y-axis is bits. It's the same meaning as in computer science. So 1 bit represents a 1 binary code, 0 or 1, and 2 bits meaning that 2 digits, 2 binary digits that can separate 4 different types of information. So that's the same meaning as the computing bits. But in this content, the information content can be represented, calculated by the Shannon entropy. Hi is the Shannon entropy at each position, and F here this means the frequency of base B refers to A, C, G, and T at the position I. So in this, after calculating these, we can calculate the whole information content of each base. So intuitively, we can imagine that if this base is more clear, it's obviously if they are very clearly to be 1 base. So for example, this position has to be G, and then we have the information content is very high, meaning that we can for sure determine that this position has to be 1 base, and meaning that they have 2 bits of information. This information can separate 2 bits, means that they can separate 1 out of the 4 possible bases. And if we want to do a practice, you can know that if you can calculate that if each of the bases are 0.25 or 1/4, so meaning that we cannot clearly separate these 4 different bases, the whole information content will be 0. And then if for some other bases, if there are 2 possible bases can occur, say for example, if some base can only be T or G, and then the whole information content will be only 1. So meaning that they can only separate those 2 bases, not the other 2. And also if the 2 bases have an equal frequency. So that's why we can observe a different heights of different positions on the whole sequence logo. And I think that's a very informative way of representing the sequence logo and sequence motif. Yeah, so this is about the background. And now I'll be talking about ChIP-seq technology of measuring chromatin epigenome. So there are many different experimental techniques to measure measure the information of epigenome, especially using next generation sequencing. So many of the technique and the related data analysis tools have been introduced in your previous few lectures. For example, RNA-seq can directly measure the transcriptome, the gene expression profile that are covered by the lectures by Dr. Goethe and Dr. Obel. And DNA-seq and attack-seq are measuring the profile of all the open chromatin or accessible chromatin regions that have been covered by Dr. Sheffield in the last lecture. So in today's lecture, we'll focus on the ChIP-seq technology, which is used to profile a specific epigenome factor or a specific protein factor across the genome. So the goal of a ChIP-seq experiment is to obtain the genome-wide profile or genome-wide positioning pattern of a given protein factor, either a transcription factor or a histone modification across the entire genome. So let's see how we do a ChIP-seq experiment. So first is the ChIP part, chromatin immunoprecipitation. So we can see, and this example is used, we use a transcription factor ChIP-seq as an example. We can see different transcription factor or different proteins will be interacting across the whole genome with the DNA across the whole genome. Each of them will have a different specific DNA binding motifs. And so first step will usually use a formaldehyde to cross-link protein DNA in vivo, so that the proteins will be tightly interacting cross-link at these DNA regions. And this is usually for transcription factor. And then we will chop the chromatin into small fractions using either sonication, usually used for transcription factor, or some enzymes, for example, micrococcus nuclease, to digest the whole chromatin. And micrococcus nuclease digestion is usually used for histone modification ChIP-seq. So after we chop this whole chromatin into small pieces, we can use a specific antibody that are specific targeting the factor we want to profile. So this antibody can only recognize the protein or the transcription factor that we want to profile, whereas they may have some off-target effect. So then we pull down, using this antibody to pull down the protein DNA complexes that in the process called the immunoprecipitation. So now we have we use the antibody to get the complexes of our interesting protein transcription factor, as well as the DNA fragments that are interacting with this with our interesting transcription factor. And next, we wash out the protein from the samples to purify the DNA. So now keep in mind that all of these DNA fragments are associated with the protein factor we are interested in. And now is the common step for next generation sequencing. We want to do PCR amplification to amplify the DNA fragments and to do the sequencing. So in summary, the process of ChIP-seq can be done in this process. And in this plot, using the example for histone modification, because the antibody is targeting to a histone tail, a histone mark. So from the original chromatin, we do immunoprecipitation targeting a specific factor in the process called ChIP. And after the DNA purification, we want to do additional process to the DNA fragments in order to do the sequencing. As most of the next generation sequencing technology, especially the Illumina sequencing technology, usually no matter how long, well, the entire DNA fragments may have various lengths, but the Illumina sequencing can usually sequence only the end of that DNA fragments, either single end or pair end. If for a single end sequencing, they can only randomly sequence one end from either end of that DNA fragments, or for paired end sequencing, they can sequence both ends simultaneously. So now let's go through review some history about the development of these important biochemical technologies. So the first cross-linking experiment was done in 1984, 37 years ago. So by Professor John Lees from Cornell University. And he found this way to use UV cross-linking that can cross-link the protein-DNA interactions in vivo. And they first did the experiments in E. coli. And a few weeks ago, you just heard the lecture by one of John Lee's former students, Dr. Mike Gerton. And then in 1993, several scientists in Germany found a way to do immunoprecipitation. So after cross-linking, they can use antibody to detect what the occurrence of certain proteins interacting with DNA that they are interested. So that's the first immunoprecipitation experiments published in Cell in 1993. So in order to do this protein information or immunochip information, get this genome-wide, that will need the technological advance or the innovative technologies after the genomics in the genomics era. So initially, this can be done using a technology called chip-chip, meaning that the first chip stands for chromatin immunoprecipitation, similar as what we described. And the second chip means a microarray. So very few people are still using microarray technologies to do genomics studies. But back in 20 years ago, the microarray technologies was the cutting edge and very state-of-the-art technologies for all the genomic studies. So for microarray, we need to pre-design a sets of probes in an array and use hybridization to detect the signal. So whether in each probe, whether in each well represents each probe sets, and then after hybridization, if your input sample has the DNA sequence that match these probe sets, they will show up the positive signal, and they can show different type of DNA, including the chip-enriched DNA or unenriched DNA, and then after the data processing and data analysis, we can know across the whole genome the location of the transcription factor or protein binding sites. So the first chip-chip experiment was designed and developed by the Richard Yang group from MIT. And the first author is Dr. Bing Ren, who is currently running a center for epigenomics at University of California at San Diego. And they are both pioneers in the epigenomic studies. So as I just described, there are obvious limitations for the microarray technologies. So one important limitation is that you need to have pre-designed probe sets to probe to the target DNA. It's not a completely unbiased way to detect the whole genome, not automatically. Of course, you can design the probe sets like tiling array techniques, meaning that you can design the probe sets across the whole genome, and that's also very expensive, not very efficient. So, but that was the technology at that time. So then after a few years, once the next generation sequencing, or especially the Illumina sequencing technology, was developed in the year around 2005 and 2006. People soon realized that one can use the next generation sequencing technologies to detect the protein DNA interaction, and they can generate the profile across the whole genome. So at that time, there are two works as the pioneering work that developed the ChIP-seq technologies. One is published in Cell by Keji Zhao group at the NIH, and the other was published in Science with the co-corresponding authors Rick Myers, which he is also an early pioneer in modern human genomics, as well as Barbara Waltz from the Caltech. Richard Myers, at that time, he was still at Stanford, I believe. And these two papers were published almost back-to-back, a few weeks apart. And usually these two papers are considered the first papers that developed ChIP-seq technology. And they both give the name of the technology called ChIP-seq. However, sometimes later, people realized that there is another work that actually published a few months earlier than the other two papers in Nature. That's a few months ago, a few months before the other two papers were published. Frank Pugh lab from Penn State, they published the paper in Nature. They use next generation sequencing to study the genome-wide distribution of a histone variant H2AZ in yeast, and they didn't give it a name called ChIP-seq, but the idea is essentially the same. They use immunoprecipitation to capture the H2AZ nucleosome, and then to use next generation sequencing technology to sequence the DNA fragments associated with the H2AZ associated nucleosome. So fairly speaking, this was actually the first ChIP-seq, or using sequencing technology to do to study the protein DNA interaction. So so that was an exciting time that a lot of groups are working towards this end, applying at that time, the newly developed next generation sequencing technologies, apply them to study epigenomics. So as we can see, these are the few first papers, earliest papers published in different high impact journals. Frank Pugh's paper published in Nature a few months earlier than the other papers, but they did the experiment only in yeast. And the Zhao group and the Barbara Waltz group, they almost developed this back-to-back, published in just around a week, a few weeks from each other. And they, so in the Cell paper, or there were 7 co-first authors on that Cell paper, and they generated the ChIP-seq profile for 20 histone modifications, as well as H2AZ and some other proteins, including POL2 and CDCF, in human CD4 plus T cells. So that was the first histone modification ChIP-seq, also the first ChIP-seq studies done in primary human cells. On the other hand, the Johnson paper published in Science, they generated one ChIP-seq profile for one transcription factor in the human cell line. And then a few months later, another paper published in Nature that led by Eric Lander and Brad Bernstein at the Broad Institute, and they published the first ChIP-seq profiles in mouse tissues, mouse cell types. And all of these papers are very highly cited and becomes the classic literature in the field. So since the development of ChIP-seq technology in 2007, there have been an increasing amount of data, ChIP-seq data published by the labs across the world to study different factors in different systems. Just for human and mouse, we can see tens of thousands of data have been deposited into the public domain for the for the past years, and the ChIP-seq has really become a predominant method for profiling epigenomes. And using the data from a public database called Systrom Data Browser, I took a look at the the number of published data for the 6 histomodifications, most common profiled histomodifications, and we can see an interesting pattern that although all of these marks are also the data are increasing, H3K27 acetylation is one mark that that are increasing very rapidly. If you still remember, H3K27 acetylation is the mark for active enhancers, and that may reflect the interest of the field of the community that are very interested in the mammalian enhancers in the genome. Okay, so this is an overview of the ChIP-seq technique, and I will briefly go through the data analysis for ChIP-seq. So the strategy for analyzing ChIP-seq data is we want to start from the raw data coming out of the sequencing machine, and the end product will be the information of protein-DNA interaction across the genome. So that's the ultimate goal for analyzing ChIP-seq data. So in the beginning, keep in mind that our goal is to analyze where across the genome is some protein factor, some transcription factor are located. And in the ChIP-seq experiment, we obtain the DNA fragments that are associated with that protein target. And we sequence the end of these DNA fragments. So this is the raw data from ChIP-seq, or actually from any Illumina sequencing experiments. And we can see it's almost unreadable, although we know that there are 3 or 4 lines represents each sequence read, and in each experiment, in each experimental each samples, there could be tens of millions or even hundreds of millions of such short sequence reads. And so in general, the strategy would be to realign these short sequence reads back to the genome to form these, if we aligned and piled up these sequence reads back to the genome. This is a snapshot of a certain region in the genome, in the genome browser. As we can see, blue and red represents the DNA fragments. I mean, the sequence reads from different strands. A certain region will have an enrichment, right? That could indicate that this could be a protein DNA interaction site or transcription factor binding site. But this type of a plot is visually is not very intuitive, and we want to convert this type of a representation to other type of representation, like this profile, which looks much nicer. So there are several questions we want to address during this strategy, during this analysis of ChIP-seq data. That includes, so first we want to find where in the genome do these sequence reads come from, meaning we want to do sequence alignment and the quality control. And the next question is, what does the enrichment of this sequence mean? Like these enrichment of the sequence in a certain region of the genome, what do they mean? So we want to we want to use p-coding to identify those enriched regions. And then finally, what can we learn from this data? That means we want to do downstream analysis to make sense of the data. Okay, so we would start from a very beginning. So the base ChIP-seq analysis, the pre-processing of the data includes the first step is using very widely used bioinformatics tools, including Bowtie2 or BWA, to do the sequence alignment, which means we want to map the short sequence reads back to the reference genome. And as we can see, both of these tools have in the name contains the word B and W, which actually stands for Boros-Wheeler algorithm. It is a very powerful algorithm for data compression and in sequence alignment. It can these algorithms both use this Boros and Wheeler transformation to make a very efficient sequence alignment. Say, if imagine that we want to, the goal is to align tens of millions of short sequences to the entire human genome of 3 billion base pairs. So it got to be a very efficient algorithm to do that very quickly. So a general rule, well, intuitive rule, although many of the advanced method is no longer using that strict cutoff. So imagine that if a sequence read cannot map to the reference genome, we have to discard it. There could be that this just sequencing error from the sequencing machine, or in a very tiny chances that could be biologically meaningful, meaning that this short sequence it actually includes some variant from the reference genome. But if they completely cannot map, we'll usually discard them. For those variants that only have one or few base nucleotide differences, say a SNP site, all of these algorithms can actually allow certain type of mismatch. And another case is that if a sequence reads can map to multiple positions in the genome, usually a repetitive regions in the genome. And then many years ago, the old algorithms simply discard this type of sequence reads because we cannot determine the only deterministic position of that sequence reads. But recently, the recent algorithms, they can give a relatively low score to these type of cases. And then what we like the best are those sequence reads that can map to a unique location in the genome, can uniquely map. So, and the next step will be the redundancy control. The redundancy control is an important step in the ChIP-seq and ChIP-seq data analysis, because imagine usually the ChIP-seq experiment is done in bulk cell type, bulk tissue, or the cell sample that consists of at least hundreds of thousands, usually millions of cells. So the signal comes from a large cell population, and in each cell in a given location in the genome, if there is such a transcription factor binding in this position across the genome, and then we use either sonication or amylase or the enzyme digestion to digest that part of the DNA, it's highly unlikely that across different cells, the cut site, the digester site, is at the exact same base pair locations. So most likely, so these are piled up from different cells, if you can imagine. So the end or the cut sites should be slightly different, right? They should be enriched in a relative narrow but a region, distributed across a region in this specific location. It's very unlikely that many of the sequence reads have the exact same base pair coordinates. So that start from the exact same base pair and end at the exact same base pair. So in this case, we usually call them a redundant reads, because these type of the scenario is most likely coming from DNA PCR overamplification, meaning that we overamplify the starting material. We didn't have enough material from the ChIP DNA, but even if we added them, including them all, it doesn't really give us any additional information for the TF binding sites. So we keep those non-redundant reads, but ignore the redundant reads. So before the peak calling, there's a few more steps that need to be done. Includes this is for the early stage of a ChIP-seq technology, which we usually use a single end sequencing, meaning that for each DNA fragment, we can only sequence either end of the whole fragment, but not both. So in this case, we want to retrieve the whole DNA fragments from the end sequence reads. So there are several approaches, two types of approaches. One is to design a peak model that is used in one of the most commonly used ChIP-seq analysis tool, MAX. Is that so for any TF binding sites, we can see the forward strand and reverse strand reads can pile up around these binding sites on both sides, on the two sides. And then they can form two peaks. So the forward strand reads can form one peak, and the reverse strand reads can form another peak. And the distance between the peak summits, the summits of these two peaks, can be estimated as the average fragment size of the entire DNA fragments. So meaning that this is the most possible position of the start 5' start position of the DNA fragments, whereas this is the most possible position of the 3' end side of that DNA fragment. So this distance is the most possible DNA fragment size. So it's this model is very powerful in identifying the especially transcription factor ChIP-seq data for estimating the DNA fragment size. But for histone modification ChIP-seq data, it's not that easy to find a very sharp information to form this peak model. Instead, one can calculate the cross correlation between all the forward strand reads signal and all the reverse strand reads signal. And the cross correlation curve is represented here, and the peak on the cross correlation curve can be regarded as the estimated fragment size. So intuitively, this means that if we observe across the genome, not necessarily in the peak region, if we observe a forward read, where in the flanking regions do we most likely to observe a reverse strand read? So that means the peak of the cross correlation curve. So this is another way of analyzing or estimating DNA fragment size that used by another commonly used ChIP-seq analysis tool, CISR. And then after we estimate the DNA fragments, we can just extend the reads to retrieve the whole DNA fragment, and then pile up the whole DNA fragment to form this peak-shaped profile. And then by having this peak-shaped profile, we can do the peak detection, peak calling or signal detection. So of course, if we use paired-end sequencing, in which each fragment has clearly known information from both ends, we don't need to do this DNA fragment estimation step. So the next is to do peak calling, meaning that we want to identify the peaks or identify the signal enriched regions across the whole genome. So as we mentioned repetitively, transcription factors are different from histone modifications. Transcription factors have a very specific, recognize very specific DNA sequence. So they usually form a very sharp peak. So they can very precisely locate it at some location in the genome across different copies of from the cell population. So they can form a very sharp peak. Whereas histone modifications, the fundamental units of histone modification is the nucleosome, right? But the nucleosome are not necessarily always positioned at a very high base pair resolution position. So in many cases, histone modification will form a very broad enrichment regions, like broad peaks. They have shown on the ChIP-seq data, they show very diffuse signal. So for sharp peaks and broad peaks, of course, we want to use a different algorithms or different bioinformatics tool to do the peak calling. So for sharp peaks, MAX is one of the most commonly used tool. They use a dynamic background to build up the model and use a Poisson to identify the highly enriched peaks. Whereas for broad peaks, a powerful tool is Sizer that I developed many years ago when I was a graduate student. They used a spatial clustering of local weak signal and to aggregate the weak signal together and to form a very broad enrichment domain, and also use a Poisson model to determine the statistical significance. So this is an example of a typical sharp peak and typical broad peak. So I would say most transcription factors are sharp peaks, and most histone modifications are forming broad peaks. And this is an example of the results that are called peak calling from Sizer. As we can see, we can form a very broad peak from the data. So I think because of time, I will skip the technical details of these algorithms. But in the sense is that MAX is used to call sharp peaks, whereas Sizer is used to call broad peaks. And for usually for both MAX and Sizer, we need to, when doing the data analysis, the important parameters that need to be we need to pay attention include those parameters shown on screen. I'm not going to go to the details of those. You can certainly find the information from these slides or from the manual when you really analyze ChIP-seq data. And the data formats that, well, if you are dealing with ChIP-seq data analysis or look at the results from ChIP-seq data, that you might see different type of data, different formats of data. So FASTQ is commonly used formats to store, to represent raw sequences. And for genomic, for most epigenomic data, they represent a certain coordinates or a certain region in the genome. So the region type of data, the most commonly used data formats for region type of data is called BED. The BED format data are also very intuitive. That represents that give you for each line represents one region in the genome, and that include the information of which chromosome this region is coming from, and the chromosomal coordinates of these region, start position, end position, as well as some score names, some scores, and a optional information of strand, plus or minus. And some other data formats that you probably will see include SAM and BAM can store aligned sequence reads information. And as for data visualization or the processed data, including the formats bed graph, or WIGO, or BigWig, they represent the pileup profiles that they can use for browser visualization, like these for ChIP-seq data. So the data process also related to different data format transformation, or during the data analysis will be like starting with the raw sequence reads from FASTQ data, and use sequence alignment tools that I mentioned with the reference genome information to have the aligned reads information. All the aligned reads that will have the coordinates of the reads in the genome in this format, and we use Max or Sizer to call peaks and to get the both the profile for visualization as well as the peak information from the genome. And I will skip the QC part. So each step in the process will involve in some quality control that if you are actually working on some data analysis, you need to pay attention, but we'll skip today. So as a summary, the experimental procedure of ChIP-seq, and I plot the data computational analysis or data analysis procedure of ChIP-seq data is essentially the data analysis or bioinformatics analysis is a trace back of all the each different steps in the experiments. So start from the very end product from experiments, which are the starting point of data analysis, that are the raw sequence reads. And each step of the analysis can be mapped to or can be matched to a certain step in the actual experiments. So the QC or quality control at each step of the computational analysis actually reflects the quality of the data, reflects the quality of certain step in the experimental procedure. So after all, the end product of the bioinformatics analysis will reflect the actual biology information from the very beginning. So that's another way of thinking computational biology or bioinformatics analysis is a way of traceback to retrieve the useful information from the every steps in the experiments. So briefly review the ChIP-seq data analysis that will include sequence read mapping, sequence alignments, and the peak calling using Max or Sizer, quality control, DNA fragment size estimation for single-end sequencing, piled up profile generation, and the peak detection. And next, having the transcription factor binding sites or histone modification enriched regions available from a ChIP-seq experiments, we need to do downstream analysis to make sense of the data, to find the real function of the data. So only having the information of where these transcription factor binds is not it's not enough to explain biology. So I will just simply go through some of web-based tools and web resource for you to do some of the downstream analysis and the integration, but I'm not going to go to the details of those. So Galaxy is a bioinformatics analysis platform that integrates a lot of different computational tools for analyzing the sequencing, next-gen sequencing-based data and the genomic data. And you will find a lot of useful tools from this Galaxy websites. And established on top of the Galaxy platform, there's a analysis pipeline, analysis platform called Systrom. This Systrom AP is also a web-based tool for you to run ChIP-seq analysis, especially Max that has been installed. And in this tool, you can do the peak calling using this web-based tool. For Sizer, you can just go to the website to download and run this in a command line manner. And then for downstream analysis, let's say data visualization, we usually visualize the data on the genome browser. There are several options for genome browser. You can browse. UCSC genome browser is one of the oldest one, used by many consortia. And the WashU genome browser is especially powerful for large-scale DNA methylation, as well as I think 3D genome interaction type of data. Say high-C data can be browsed. It's very powerful at WashU genome browser. And IGV is genome browser that one can install on your own laptop, developed by Broad Institute. It's a very convenient and efficient genome browser. So one important function or one important work to associate epigenome information with gene expression is to do integration, right? We want to find, we want to associate the epigenome signals to the nearby genes. So one tool I might want to mention here is called beta, binding expression. I forgot what that stands for. Yeah, binding expression target analysis. So they used a summarized approach in which they start from each gene. They summarize all the epigenome signals that in the not around this gene in a fairly far region to include the distal enhancers and give it the distance weight so that they can assign a score to each gene that represents how many and how close the epigenome signals, TF binding sites or histone modification binding sites, are close to this gene, so that you can directly associate these score with the gene expression to do integration analysis. And several other epigenome data integration tool and analysis tool, or just a list here for you can go ahead and browse these websites to see what it does. Some useful data resources includes the ENCODE. ENCODE is a large consortium that generates a lot of data capturing epigenome informations. So the ENCODE stands for Encyclopedia of DNA Elements. The goal was to profile or to measure the function and the information of as many DNA regulatory elements or DNA functional elements as possible from a few model systems, including human and mouse, and also worm and fly. So they have a lot of high quality various type of DNA epigenome data, including ChIP-seq data. And if you are very specifically interested in ChIP-seq, so there is a database called System Data Browser, which is one of the most complete data collection for all publicly available ChIP-seq data for human and mouse, focusing on high quality transcription factor and histone modification data. Say if you are interested in some transcription factor, or you want to see what cell type can we find these ChIP-seq data for this factor, you can browse this system data browser to find those data analysis. Or if you want to design a ChIP-seq experiment and you don't know which antibody to use, you want to find a high quality antibody, and then you can browse this browser to collect all the ChIP-seq data for that specific factor that you want to profile, and look at the quality control of those data sets and select the antibody that was used in others' data to generate high quality data. So that's a very useful and powerful tools for to collect and apply and use public available information. So I think I will skip this. So this is a bioinformatics tool that my lab developed for the past few years, in which we want to take advantage of these collected tens of thousands of ChIP-seq data to do functional predictions of transcriptional regulation, in which you can use the gene sets or other type of data as input, and the output, the BART tool will be able to tell you which possible transcriptional regulators or transcription factors are regulating the input gene sets or associated with your input genomic features. Okay, so I think that's a review of the data analysis part. We just mentioned the downstream analysis integration, and I would encourage you to take advantage of the public available resources. Finally, I want to briefly mention some future perspectives. So ChIP-seq technology has been developed for nearly 14 years, and they are used primarily to study, they need to use a large amount of cell to study the general genome profile of the epigenome, but there are certain limitations of ChIP-seq. Of course, first of all, it highly depends on the availability and the quality of the antibody. So for example, in the System data browser databases, for human, they have collected over 900 transcription factors. But in the entire human genome, there are about 1,600 transcription factors. The remaining them is very possible reason that we still don't have ChIP-seq data is because we don't have a high quality antibody to profile them. So that's the availability of antibody is one of the main limiting factor of studying comprehensively studying all the epigenome information. And the second point, well, ChIP-seq is I will only say this is a semi-quantitative approach. It's not a completely quantitative. So one reason is because during the experimental procedure, there are the experimental protocol is fairly complicated, and it's very difficult to make the quantitative control, precise quantitative control on every single step. And usually, ChIP-seq, when doing a comparative or differential analysis for ChIP-seq across different conditions for different cell types, ChIP-seq is as a genomics profiling technique. It is not used to detect a global change of either transcription factor or histone modification patterns. Say in some cases that you may want to perturb the histone modifying enzyme to see whether there are across the genome there are global increase or decrease of some histone modification. But ChIP-seq is not the technique to detect the global increase or decrease because of the technical limitations. And I mentioned that ChIP-seq needs many cells, usually millions of cells. So sometimes it's difficult for example, for primary cell tissue or especially for clinical samples, the amount of cells is limited. And because ChIP-seq is done in a large population, in a large sample, it cannot consider the cellular heterogeneity. It's an average, the signal is an average across a large cell population. So I will briefly mention there are several other techniques that are usually a variant of ChIP-seq or other genomic technologies that can also measure the genome-wide distribution of this epigenome factors. ChIP-exo and ChIP-nexus are both variants of ChIP-seq. Actually, ChIP-nexus is further developed from ChIP-exo. These two technologies are used to generate a very high resolution profile, usually up to a single base pair resolution profile of transcription factor binding pattern. And ChIP-exo was developed by Frank Pugh, the one who also published one of the earlier ChIP-seq work. And ChIP-nexus from Julia Zettlinger was also a former lab member from Richard Young's lab. Achievement and the cut and run and cut and tag, these technologies are used especially for smaller cell samples. So if the sample have much fewer cells than the usual ChIP-seq can deal with, these technologies can deal with generate epigenome profile from relatively fewer cells. And I'll just put the name here, but don't mention any details about the recent years. There's a breakthrough or a lot of technological advances on the single cell genomic assays and the spatial genomic assays that can gradually profile the epigenome information on the single cell level or even with this additional spatial single cell resolution. So that's some even more advanced and future technologies are being developed. So as a final summary, in this lecture, I told you that about the transcription factors and the histone modifications, which are two categories of functionally important factors in the epigenome. And we mainly focus on the technique ChIP-seq, which is used to profile protein DNA interaction informations in the epigenome. And for ChIP-seq data analysis, data processing, specific for ChIP-seq, the specific type for ChIP-seq analysis are the peak calling, and I briefly mentioned the two peak calling tools, MAX and CISR, for narrow and broad peak calling respectively. And I want to also give the information and resources about other online tools and resources in which I encourage you to browse, and hopefully they can be helpful to your future research in the genomics and epigenomics studies. So I'll end here. Thank you very much.